Much of the content of this course discussed what we do and do not know about the history of the Earth and the life it contains, as this is our only known example of an origin of life. There have been many recent advances in the understanding of early Earth history, and as technology improves, this understanding will continue to be refined. I would like to leave the course with a summary of content by highlighting some of the open questions scientists are still working to understand within the origins of life umbrella. For example, in what environment did life first emerge? We presented many models and possibilities. However, our atmospheric and geological models of early Earth are still only loosely constrained, perhaps too much so to draw definitive conclusions of land masses, water sources, and continental and atmospheric compositions, especially when the window for life's emergence is 500 million years long. Even if we could constrain the time and the general environmental conditions under which life formed, the Earth would still have many local environments and climates that could be extremely important to the formation of life, for example, hydrothermal vents. We also discussed what we think early life may have looked like, from the RNA world to the formation of protocells. From the content provided, it should be obvious that scientists are approaching this idea from many angles with little convergence, leading to our next open question. What were the components of early cells? And what parts of life came later? We discussed in depth many of the extant biological molecules and possible prebiotic syntheses to produce some of these compounds. We also demonstrated computational models that can be used to understand how simple mixtures can evolve patterns, maintain autocatalysis, and model informational complexity. Additionally, we discussed the last universal common ancestor, or LUCA, and possible precursors to the portions of LUCA's already complex metabolic and genetic components. However, many parts of the earliest cells are still a matter of debate. For example, did these earliest cells use RNA? Did they have cell-like membranes? This lack of understanding makes modeling such systems challenging. Another open question is, will we be able to find life elsewhere? When we turned to astrobiology, we discussed where we are looking for signs of life elsewhere in the solar system, how we might identify places that harbor life, and what that life might look like. There are many challenges of finding life elsewhere, especially if it is not within our own solar system. While Mars and some moons are promising for finding life, or the remains of past life, if a living system is very different from Earth life, we may not even recognize it. Additionally, issues with contamination and planetary protection, both of our world and theirs, only adds to the challenges of finding and understanding life elsewhere. That brings us to our final question, perhaps the biggest of all. What is life? Is carbon-based life the only example of life? We talked in depth about how models can be used to understand living things, predict behaviors, and model events. Additionally, we presented many of the recent strides theory has made in defining properties of life that we know are controlled by existing mathematical models. However, scientists only have a working definition of life without a general theory or model for understanding living processes, and it is difficult to create a definition of life that is not based solely on our singular instance of life. We would like a general theory and mathematical definitions that can be used to better define how, if, and when non-living material transitions to living material. If we develop a general theory of life, it still may only be possible to confirm such a theory when a second instance of life is known. Thank you for your participation in this course. If you'd like to help support future content like this, please consider donating to Complexity Explorer.